This program demonstrates some of the things that can be done with DNA in what we call molecular biology and genetic engineering. It had long been known that DNA carries the genetic information, but it was not until Watson and Crick's discoveries in 1953 that the importance of the DNA double helix was realized. Later, the processes of protein synthesis and DNA replication were unraveled. By the early 1980s, some biochemists, who were then starting to call themselves molecular biologists, were beginning to find ways of manipulating DNA. This meant actually altering the genetic material of cells. The manipulations were carried out using enzymes that act on DNA. Soon it became possible to determine the sequence of bases in DNA, the genetic message itself. In this program, you will see how a long, double-stranded molecule of DNA can be cut using restriction enzymes, how the lengths of the pieces can be measured by electrophoresis, how a length of DNA can be amplified using a special reaction called the polymerase chain reaction, and how a piece of human DNA, in other words a gene, may be cloned by inserting it into a bacterial cell. Most research involving DNA manipulation is carried out on the micro scale, using microliters of liquid rather than milliliters or liters. This involves using specialized equipment like this. There are a number of reasons for working on the micro scale, one of which is that many of the reagents used are very expensive. This is a micropipette of the type commonly used in biochemical work with DNA. It has an adjustable micrometer plunger arrangement that allows it to deliver any volume between 10 and 200 microliters. The micropipette is used with a plastic disposable tip. Unlike glass, water doesn't cling to plastic, so we don't have to worry about droplets on the outside or about draining times. The tips we are using have been sterilized in an autoclave. This is because many microscale experiments must be performed under sterile conditions. We don't want bacterial DNA or enzymes contaminating the experiments. After use, the tip must be discarded into an appropriate container. This is particularly important if it has been used to pipette radioactive materials, bacteria or viruses, all of which are often used in molecular biology. We need other microscale equipment to go with the micropipettes. These are plastic microtest tubes and a microcentrifuge. This is just about all the equipment we need to start doing molecular biology work on a micro scale. The first experiment we're going to see shows how to cut a double-stranded DNA molecule using a restriction enzyme. In order to explain how this works, we need to look very closely at a short section of DNA. It will be easier to see what is happening if we straighten the double helix out. The experiment uses a restriction enzyme extracted from bacteria to cut the DNA sequence in a specific place. A different restriction enzyme would cut the DNA in a different place, but each cuts at a specific sequence of bases in double-stranded DNA. The enzyme we're going to use is called HINF1. It comes from the bacterium Haemophilus influenzae. Note that the cut is not at the same place in each DNA strand, so that when the strands are pulled apart, they leave an overhang at each end. Here is how we carry out the cutting in the laboratory. A few microliters of a solution containing DNA is placed in a little plastic test tube. This contains about 50 nanograms of DNA. Ten microliters of a buffer solution is added to keep the pH constant. Now for the restriction enzyme. 1 microliter is all that is required. When this has been added, the tube is closed and the contents mixed briefly using a whirly mixer. A quick spin in the centrifuge brings all the contents to the bottom of the tube. This is then transferred to a water bath, where it is incubated at 37 degrees centigrade for 10 minutes. This allows the reaction to take place. At the end of the incubation period, we add a little of a compound called EDTA, which stops the enzyme reaction. Then we add some blue dye. This will act as a convenient marker so that we can see what we are doing in the next part of the experiment, which is to analyse the DNA by electrophoresis. This will confirm that the DNA has indeed been cut, and will also tell us how long the segments of DNA are. Electrophoresis is a gentle way of separating or analysing biological molecules such as DNA and proteins. It works by separating molecules from each other on the basis of charge. Positively charged molecules move towards the cathode in an electric field, and negatively charged ones move towards the anode. In addition, molecules with a high charge move more rapidly than those with a low charge. Size is important too. Large molecules move more slowly than small ones. Now, let's look at that last sequence again. See if you can spot what is happening. Imagine that this is a mixture of molecules of different sizes and different charges. We will now demonstrate the experiment using this electrophoresis apparatus and an agarose gel. We use a gel because we want to separate not only by charge but also by size. The gel forms a sort of sieve that lets small molecules of DNA move more rapidly than large ones. Agar is a polysaccharide obtained from seaweed, which when dissolved in water forms a gel. Agarose is a highly purified form of agar. The gel is made by putting a measured amount of agarose powder into a test tube and adding buffer. This mixture is heated in a boiling water bath until all the powder has dissolved. The liquid gel is then poured into a flat mould and allowed to cool. There is a plastic comb called a former standing over the dish at one end, with the teeth of the comb sticking into the gel. When the gel has set, this is removed. The teeth leave slots in the gel into which the samples will be placed. The experiment is performed using this Perspex electrophoresis tank. It has a reservoir at each end for buffer solution and a raised central section on which the gel is placed. There is a platinum electrode at each end of the tank. The red spot identifies this one as the positive. If we look closely, we can see the thin platinum wire electrode embedded in the Perspex. The first step in the experimental procedure is to fill the tank with buffer solution. The slab of gel can now be placed in position. First, we remove the sticky tape that held the gel in the mould while it was setting. It can then be put into the electrophoresis tank. In a moment, we will put the DNA samples in and start the current. But before that, we will look at some diagrams of how the electrophoretic separation works. Remember that DNA, deoxyribonucleic acid, is negatively charged because of all the phosphate groups on it. For every base there is one phosphate and therefore one negative charge. Consequently, large DNA molecules have a higher charge than smaller ones. In electrophoresis, all will move towards the positive electrode. When we carry out electrophoresis in an agarose gel, the small DNA molecules can move rapidly, while larger DNA molecules are held back by the gel, which acts rather like a sieve. So we get a separation that is based on size. Small molecules go a long way in the gel, medium-sized molecules a moderate way, and large molecules only a short distance. This is a very convenient way of finding out how many base pairs there are in a piece of DNA, in other words, how long it is. The experiment takes about an hour to run and is very sensitive. We can work with 1 microgram of DNA, or even less. Now let's see the experiment. These are the samples of DNA we're going to run in the gel. Each sample is carefully pipetted through the buffer into a slot in the gel. A steady hand is needed at this stage. When all the samples have been loaded into the gel, the lid is placed on the electrophoresis tank. It is designed so that the electrodes cannot be touched when the current is on. After connecting the positive and negative wires to the power source, we switch the unit on and set the voltage to 100 volts. After an hour or so, the electrophoresis is complete. You can see that the blue marker dye, which moves faster than any of the DNA molecules, has moved quite a long way. However, you can't see the DNA itself. We need to make it visible by staining with a dye, called ethidium bromide. This fluoresces when exposed to ultraviolet light. Now that the electrophoresis is finished, we can switch off the current and remove the lid. The next step is to stain the gel with ethidium bromide so that we can see the bands of DNA. After staining, the gel is placed on an ultraviolet light source which has a camera mounted above it. It's very important to wear a visor because ultraviolet light causes serious damage to skin. When we turn the room lights off, we can see the DNA bands. This is the positive end of the gel, so these are the smaller molecules. This track is of a sample of DNA that has molecules of different sizes. It contains the DNA that was cut using the restriction enzyme. You can see that several pieces have been produced. When a cell divides, its DNA must be duplicated. Each strand of the DNA double helix acts as a template for making a new strand. Each daughter molecule of DNA contains one parental strand and one newly made strand. This process is called semi-conservative replication of DNA. The main enzyme involved is DNA polymerase. This enzyme can take a single-stranded piece of DNA and, with a little bit of primer, make a double helix. It does this by adding new base units that are complementary to those in the original strand. In this way, a new double helix is formed. This suggested a way in which specific bits of DNA could be amplified. This reaction, which is carried out in the laboratory, is called the polymerase chain reaction. Using it, we can select a section of DNA from a long length and amplify it 1 million or more times. You might ask why anyone would want to amplify a piece of DNA 1 million times. Well, there are several reasons. One is to isolate a gene that we might want to place in a bacterial cell. One example could be the gene for human insulin. You might want to detect a gene, perhaps in an individual carrying a mutation, such as sickle cell anemia or phenylketonuria. The band of mutated DNA might be detected on an electrophoresis gel. You could start with a very small amount of DNA, say that contained in just a single cell. We are now going to see how the polymerase chain reaction is carried out. At the end, the products will be analysed by electrophoresis on an agarose gel. The starting point is having short lengths of DNA called primers. These are manufactured chemically on a DNA synthesizer, a gene machine, like this one. These 4 bases will be added together in a computer-controlled sequence to make up the primer. The synthesizer can be programmed to produce primers that match up with specific sequences in some DNA from a cell. We need 2 primers, one for each DNA strand, that between them bracket the section of DNA we wish to amplify. The other thing we need is the DNA polymerase enzyme itself, Taq polymerase, because we're going to make more DNA. We will go through one cycle of the process slowly, so that you can see what is happening. This is the piece of double-stranded DNA we want to amplify. The gauge on the right will show the changes in temperature that are an essential part of the procedure. First, we raise the temperature to nearly boiling. This separates the 2 strands. Now we add the primers, the short bits of DNA, and cool slowly to 56 degrees. The primers find the stretches of DNA to which they are complementary and anneal to them. Next we add the DNA polymerase, represented here by the green circles. Remember that in order to make more DNA, this needs single-stranded DNA plus a primer, which is exactly what we have here. The polymerase operates best at an intermediate temperature, so now we heat the mixture to 73 degrees. The enzyme creates a second complementary strand of DNA along each of the single strands. If these temperatures seem rather high for living organisms, you'll see why they're used in a minute. We now have 2 lengths of double-stranded DNA, where we had 1 before. The whole process is then repeated. Heating to 95 degrees separates the 2 double strands into 4 single ones. We don't need to add more primers because they were originally present in great excess. So when we cool the mixture, spare primers will anneal to each of the 4 strands. The large number of primers also makes sure that the original strands are unlikely to find each other again. They are always more likely to pair up with a primer. We don't need to add any more DNA polymerase either, so when we reheat to 73 degrees, this starts working again automatically. The polymerase we're using is not adversely affected by high temperatures, since it originates from a bacterium whose natural habitat is hot springs. This remarkable enzyme survives heating to 98 degrees and works quite effectively at 73 degrees, so we don't have to add new enzyme at each cycle. Now we have created 4 double strands of DNA from the original one. We can continue into the next cycle, heating to separate the strands, cooling to allow the primers to anneal, reheating to encourage the action of the DNA polymerase. So now we have 8 double strands of DNA. The next cycle will produce 16, the next 32, and so on. 30 or 40 cycles will produce millions of copies of the original length of double-stranded DNA. Now we are going to see this happening in the lab. This piece of apparatus is called a thermocycler. It has heating blocks, the temperature of which can be precisely controlled. The sample tube is placed in one of the blocks, and the appropriate cycle of time and temperature is set on the computer keypad. This tube contains the heat-resistant DNA polymerase. It is the last active ingredient to be added to the sample. We then fill up the tube with some light oil. This floats on the top of the other contents. It stops evaporation and ensures good heat transfer without affecting the procedure. It will be removed at the end. The computer can now be instructed to run the appropriate program. At the start, the strands of DNA are separated by raising the temperature of the block to nearly boiling. Then the temperature will be lowered to allow the primers to anneal with the separated strands. 56 degrees for a minute or two is long enough for this to take place. The temperature is then raised to the point, 73 degrees, at which the DNA polymerase works most efficiently. It will add several thousand bases in a few minutes, a remarkable enzyme. That is one cycle completed. Over the next few hours, the machine will run through 30 cycles automatically. 95 degrees, 56 degrees, 73 degrees, and so on, until the program is complete. Now we take the tube out of the thermoblock and remove the oil layer. The next step is to analyze the sample by electrophoresis to see if any DNA has been amplified. We don't need all the sample for the analysis, so we take a few microliters off into another microtest tube. This is all we need to carry out the electrophoresis test. To make the sample visible on the agarose gel, a blue dye is added. The liquid is sucked back into the pipette and expelled again a few times to ensure that the dye and the sample are thoroughly mixed. You saw the electrophoresis procedure earlier. The samples are pipetted into slots in the agarose gel, and the voltage is set at 100. After staining with ethidium bromide, the gel can be analysed on the ultraviolet light box. The fluorescent bands show where the DNA is. The small band here is the one that has resulted from the polymerase chain reaction. Starting from nanograms or less of DNA, we now have enough to be visible on a gel, a few micrograms. Although this may not seem very much, it is quite enough for a DNA cloning experiment. The band containing the DNA can easily be cut out of the gel with a scalpel blade and the DNA extracted from it. Cloning DNA means inserting a piece of DNA into a cell, often a bacterial cell, and then getting that cell to grow and divide to produce a clone of identical individuals, all containing replicas of this piece of DNA. Each of the colonies of bacteria you can see growing on this Petri dish is a clone of identical cells. These have been derived from a single cell by repeated division. We often want our clone of bacterial cells to express the inserted gene. That is, to produce the protein encoded in the DNA. This is a way of producing large amounts of proteins for commercial or medical use. Examples of proteins now produced in this way are human insulin and human growth hormone. The technique of cloning used daily in molecular biology labs is called recombinant DNA technology. This is because the gene in question is usually combined with a small circle of bacterial DNA called a plasmid. This is an electron micrograph of some plasmids. You can see they are composed of short lengths of DNA. The modified or recombinant plasmids are reinserted into the bacterial cells. This is an easy way of inserting a bit of DNA into bacteria and getting it expressed. Now we are going to see how cloning can be carried out in the laboratory. Only the most important steps will be shown. Suppose we want to put the gene for human insulin into bacteria. We will need some plasmids, some bacterial cells, nutrient medium, and of course the gene itself, the piece of DNA. First of all, plasmids. Where do we get them from? Plasmids are small circles of bacterial DNA, a few thousand base pairs long, that are separate from the main chromosome. Each plasmid carries a few genes, often the genes for resistance to antibiotics. Plasmids can be transferred from one bacterial cell to another. This is how resistance to antibiotics can spread. In the laboratory, we cut open the plasmid circle of DNA using a restriction enzyme. You have already seen how this is done. Now we insert our gene, say the one for human insulin, and put the closed circles of DNA back into bacteria. When the bacteria divide, they will form a clone of cells, all producing insulin. This incubator contains cells of the common gut bacterium, Escherichia coli, growing in a nutrient medium. They are a rather fastidious strain of cells and cannot survive long in the absence of the special growth medium. There is no danger of their escaping from the laboratory and spreading unwanted genes in the wild. These cells contain plasmids. When we have enough bacterial cells, they are spun down in a centrifuge to separate them from the medium in which they were grown. Next, the cell walls will be digested away with lysozyme. You may know that Sir Alexander Fleming first identified the enzyme lysozyme in human tears. After the mixture of cell contents has been centrifuged to remove any fragments of cell wall, we carry out ultracentrifugation. The pink colour is due to ethidium bromide, which has been added to make the plasmid DNA visible at the end of the procedure. The tubes are loaded into an ultracentrifuge, where the plasmids are purified on a density gradient of cesium chloride. The ultracentrifuge is run at high speed overnight. This is called density gradient ultracentrifugation. Next morning, the tubes containing the plasmids are carefully removed. The bands of DNA are clearly visible. The bottom band is the one we're interested in, as it contains the plasmid DNA. To remove this, the tube must first be clamped securely. We have to be very careful at this stage not to disturb the contents. Because the tube is made of plastic, it's quite easy to remove the band containing the plasmid DNA. First, we make a little hole in the top of the tube to let air in. This will make it much easier to extract the band at the bottom. Now we insert the needle from a 2 ml syringe into the tube just below the band we want. We then simply suck the liquid containing the plasmid DNA into the syringe. Following safe disposal of the needle, the liquid is transferred to a test tube. We now add 70% ethanol. This will make it easier to remove the ethidium bromide and recover the DNA, which is precipitated by cold ethanol. The tube is sealed with parafilm and put on dry ice. This precipitates the plasmid DNA so that we can recover it by centrifugation. After the tubes have been centrifuged, the liquid is carefully poured off, leaving the plasmid DNA behind. The purity of the DNA is usually checked at this stage by electrophoresis. The band formed by the plasmid is readily visible on the electrophoresis gel. Now we are ready to insert the gene we want to clone into the plasmid circles. We're going to cut the circles open with a restriction enzyme, insert our gene, and then put the plasmids back into receptive bacterial cells. You will remember from part 1 of this video that when restriction enzymes cut double-stranded DNA, they leave overhanging ends. If our gene, the piece of DNA we wish to insert, has identical overhanging ends, it will easily fit into the plasmid circle. The plasmid is cut open with the restriction enzyme. The gene we want to insert has been cut in the same way. Therefore, the overhanging ends match up and the gene fits into the circle. The overhanging ends are sometimes called sticky ends. They match up because the base pairs are complementary to one another. Now we are ready to insert this modified plasmid into the bacterial cells. The first step is to centrifuge the cultured bacteria. You can see the cloudy suspension in the test tubes. The centrifuge is run at 3,000 revolutions per minute for 10 minutes. When the tubes are removed, the bacterial cells are clearly visible as a pellet at the bottom of the plastic tube. The liquid is carefully poured off and the pellet placed on ice. Calcium chloride solution is added to make the cells receptive to plasmids entering them. We say they are competent. The tube is tapped gently to resuspend the bacterial cells. They need to be treated with care at this stage because they are fragile. Now we add the plasmid which contains the gene we wish to clone. To make the cells more receptive to the plasmids, we need to give them a brief temperature shock. So they are placed in a water bath at 41 degrees. After a few minutes, the tube is put back onto the ice and left to stand for a while. Some fresh nutrient broth is added to get the cells growing again. Now, of course, they contain the modified DNA plasmids. At this stage, we need to increase the number of cells by growing them on agar. We put a few drops of the transformed culture onto each Petri dish. And spread them over the surface of the agar. The plates are put in an incubator overnight so that the bacteria can grow. The temperature for optimal growth is 37 degrees. Next day, we take the plates out. The individual cells will all have grown into colonies. The final step is to select the colonies we want. Now, when we put the plasmids into bacteria, the process is not very efficient. Only a proportion of the bacterial cells will take up a plasmid. We need to select those cells that have a plasmid in them. We do this by using the antibiotic resistance marker. The cells containing a plasmid are resistant to the antibiotic ampicillin. We grow the bacteria on agar plates containing ampicillin, so only those cells with a plasmid in them survive. There is a problem with the plasmids too. Most of them will not have taken up the extra gene, the circle's just reclosed, so only a few will have the gene we inserted. We need to find the bacterial cells that contain a plasmid with our added gene. We use a cunning plan to pick out the colonies derived from these cells. As you will see, they will reveal themselves by their colour. When we used the restriction enzyme to cut open the plasmid circle, we selected one that cuts in the middle of one of its genes. The gene we selected is that coding for a particular enzyme, lactase. So when we cut the circle and insert our gene, this will make the split gene inoperative. This means that any bacteria containing plasmids with the inserted gene can no longer make the enzyme. Those without an insert can still make it. So how can we detect which colonies can still make this enzyme and which cannot? The secret is to grow the colonies on agar containing an artificial substrate of the lactase enzyme, one that turns blue if the enzyme is present. You can see that some of the colonies on this plate are blue and some are white. The white ones, those that cannot produce the lactose-splitting enzyme, are the ones we want, because they must have our gene inserted into their plasmids. The final step is simply to pick these colonies off the agar plate and grow up cultures from them. These cultures should be producing human insulin, if that's the gene that we inserted. A fermenter like this one can produce millions of copies from just a few cells. This would enable human insulin, or in principle any protein, to be produced in commercial quantities. What you've seen in this program is only a little of what constitutes molecular biology today. You haven't seen DNA sequencing or cloning using bacteriophages. Cloning is now also done in cells other than bacteria, such as yeasts. Putting genes into human cells is not all that far off as a medical treatment. In the future, this will enable individuals with genetic deficiencies to be cured simply by giving them modified genes. Molecular biology methods allow us to isolate and modify the genes required for such treatments. Both the polymerase chain reaction and cloning allow genes to be selected and amplified. In this program, you have seen some of the ways in which DNA can be manipulated: cut, analysed by electrophoresis, amplified, and cloned into bacterial cells. The tools for doing this are the enzymes that deal with DNA in cells. You will have noticed that most of the operations are carried out on the micro scale and are actually rather simple. The methods of analysis have to be sensitive in order to cope with the tiny amounts of DNA to be analysed. Almost anything can now be done with DNA in the laboratory. The key is to plan the experiments that further our knowledge and lead to a better understanding of how the genetic material works.